I'm going to show you my lab space and some of the equipment that we're using for the photoacoustic calorimetry work that we're doing to identify imaging agents. And first I'll start out by showing you major instrumentation that we have in the space, and then I'll demonstrate exactly how the layout of our instrument is set up. Now here you can see the entire setup that we have in this laboratory. I'm actually inside of a biohazard level 2 lab with a BSL-3 inner shell right down there by where the garbage cans are. I don't work in the BSL-3. I do collaborate with other researchers here in our BSL-2, trying some things out that we might be able to do for some viral work and for some imaging work. Now, this is my general space that I work in. I have a few other places that I'll work in our building as well. But inside of this space, you can see that I've got a decent array of instrumentation. I have a UV-Vis double beam spectrophotometer here that we use to do absorbance measurements for our samples to make sure that we have accurate absorbances for any of these samples. The absorbance goes into the exponent of one of the terms in our equations, and so having our absorbances be really well controlled is a critical part of the work that we're doing in here. Now, we also have a fairly robust bay for rebuilding and modifying our electronics. So we have a good amount of parts there. I have a 3D printer here that we use to generate some components. You can see here that we have a positionable mount for optical benches, and there's a webcam there that we're working to get working to give us real-time observations of our 3D prints while they're going, in case that we see a problem. So we have a bunch of our solutions ready to go for the project that we're doing to find in-house created optical density filters for the laser. Now, coming along from over to there, you see here is where our oscilloscope and our computer control for that oscilloscope reside. We have a separate computer over here that controls this fluorimeter that we can use to measure if we have any luminescence for the dyes that we're working with. We have a temperature control stage for our laser system. The speed of sound in water varies with temperature, and we can exploit that in order to measure the volume change of a photo-initiated reaction. And so by manipulating that, we can find out about any of those volume changes in the reactions. Now, here is part of the control module for our OPO laser. You can see that here's the power source for it, and over here is our optical system. I've got my curtain pulled away. You can see I have a wooden frame that I built just to keep things shielded over here so we don't have to worry about stray beams while we're working. Now, here's how the instrument is set up. We have an optical parametric oscillator as our light source. I suppose technically it's not a laser, but it has a laser inside of it, and the OPO gives us whatever color we want. Continuously tunable from 400 out to, I believe, somewhere in the mid-thousands range. I can't quite remember the nanometer that it goes to. Then we have our optics path over here. Now, let me do a top view to show how this is actually set up. The light emits here. We hit this microscope slide cover that we're using just as a very gentle beam splitter. We don't want to have a thick one because then we'll have internal reflections giving us offset beams. We want to keep things fairly optically simple. So we just reflect a little bit off the surface of it over to this photodiode. This is our trigger that tells the oscilloscope when our laser is fired. That way we don't have to worry about any temporal jitter in the firing of the laser. Everything is contextualized to when the light actually passes through our system. Now we have a aperture here so we can keep the beam geometry very circular and change the size as needed. Coming back to the top view, we were just seeing our neutral density filter wheel here, so we can change how much attenuation we're giving the system. This is where we control the light setting for each of our usually 7 experiments that we do to measure F sub h of a single substance. We'll typically do 4 of those repetitions in an experiment. 2 of those will be our standards, 2 of those will be our analyte, and then we'll do a 5th of just our solvent. So 7 settings, that means a total of 35 runs per experiment. Now here we have a cubic beam splitter. This one is visible light only. If I go down to the UV, the optical cement that's on it starts to absorb, so we just don't use it down in the ultraviolet. Over here we have one of our photoacoustic cells, and you can see that we got it loaded with a blue solution of brilliant blue BN, BBBN. And so I just keep a bit of it ready to go. This serves as our real-time measurement of the laser's firing intensity. We also have a fiber optic here to monitor the actual output wavelength, along with a real-time measurement that comes from the laser itself. So between the two of them, we have a constant notion of what our wavelength is. The other thing coming out of our beam splitter, the other beam path, will be straight into our photoacoustic setup. You can see here that we have a Q-pod from Quantum Northwest that we're using. It has a micrometer so we can position it back and forth in space. I can flow gas through to make sure that it's only dry gas and I don't have any condensation when we're using cold temperatures, this being Southwest Florida and all. I can position it up and down and get it centered in the beam path as needed and put slits in if I need to. Here's where the microphone assembly is actually set up. The whole thing has a Peltier-cooled stage, or heated stage, and so we have temperature control. We can keep it controlled and constant to 2 decimal places on the degree in Celsius, and we flow the water through as part of the cooling, or at least the temperature acclimation for that. That's basically how the instrument is set up. So we send the light pulse down through, measure when it fired, adjust the intensity, real-time measurement of energy per pulse, sample position here.